do a general overview of the materials we covered on Monday's lecture for A&P. So we did the neuro lecture, and in that lecture, what we covered predominantly was the sensory system, the afferent system. And then I said I'm going to cover extensively the autonomic system, which is the parasympathetic system and the sympathetic system. And the reason why I think these are the systems that you really need to be familiar with is this system here will deal with a lot of pharmacological issues, especially medications, receptors, and how they affect the body. So if you know the normal anatomy and physiology of this system, you'll be in good hands when you start learning about the pharmacology and the effects they exert. In the musculoskeletal system, I specifically went over the somatic system and all the functions with that. So if you want to review that, please review those lectures. So today is going to be the sensory system, and it's going to be the autonomic system, which is going to be the parasympathetic and the sympathetic, right? And here I'm going to build on the concepts we learned about in the muscle chapter. And in the muscle chapter, we'll learn the propagation of the impulse, how it travels right in the axon. And the way it does it, it's causing these waves of depolarization to repolarization and sweeps across the axonal membrane. And it does so all the way down to the terminal branches here being axonal terminals. Upon reaching the axonal terminals, it will release specific neurotransmitter. In the muscle chapter, we learned the neurotransmitter was acetylcholine that's being released, and that exerts its effects on motor end plate. Today, we're going to learn with autonomic nervous system, there's going to be preganglionic neuron, which secretes acetylcholine for the most part. And for the sympathetic system, the postganglionic neuron is going to secrete norepinephrine. So it's a different neurotransmitter, and the effect it exerts differ from the acetylcholine that was secreted in the muscle cell acting on the nicotinic receptors. So this is what we learned in musculoskeletal chapter, right? We said the impulse comes in, right? And the moment impulse comes in, it causes waves of depolarization, which is mediated by sodium channels. So depolarization mediated by sodium, and repolarization is mediated by potassium. I'll just use a different color. And the other important concepts, I said that the cells are predominantly bags of potassium and a sea of sodium, and we're going to build on that. Once the impulse comes in, it opens sodium channels. There's a threshold potential that's reached, right? And we also have calcium voltage-gated channels. So the very important thing about calcium was the following. On the presynaptic neuron, this is the presynaptic neuron, right? And this is the postsynaptic neuron. On the presynaptic neuron, calcium function is to essentially bind to this vesicle, right? And cause it to dock to the membrane, right? And what it will do, it is going to cause calcium-mediated exocytosis of the neurotransmitter acetylcholine in this example. And once the acetylcholine is released, acetylcholine binds to the nicotinic receptors, or you could call them acetylcholine nicotinic receptors. Once it does that, sodium will come in, depolarize motor end plate. And calcium here, the role of calcium once it comes in here, and it's released from the sarcoplasmic reticulum, we learned that in the muscle, it was causing contraction of the muscle by binding to troponin, moving the trypomyosin out of the way, and myosin head docking to the actin site, causing that power stroke. So presynaptic site, calcium's role is to release acetylcholine, and in the postsynaptic membrane, the role of calcium is to actually cause the mechanical contraction of the muscle. And we're going to see what other neurotransmitters do a little bit in this lecture. Okay? So I talked about different types of neurons and their function. The one we learned about in the muscle, right, was this motor neuron. And the signal was propagating from node to node, right, as I just explained. And it went like that. And it went to the muscle site and it triggered the response. Now, if you look closely, this neuron, this muscle motor neuron, it has these fat cells surrounding the axon. What are these fat cells? So the fat cells, they're essentially called myelin sheath. Think of it like if you have a wire and it's insulation, right? So it prevents current from shocking you, right? If it was protecting the wire. So myelin sheath, it will insulate the axon. And what it does is it's a layer of fat. If you think about, let's say if I took like a cell and I used an iron to flatten it out, this is the nucleus, right? And I said this was the iron that I used to flatten out. And then I wrapped it around my neuron. And this fat is composed of sphingomyelin. And what it does is it insulates so that the only sites of ion exchange occur at these locations called node of Ranvier. So nodes of Ranvier is how you see the impulse propagating. Why? Because no ions can leak here where I'm drawing. Why? Because it's surrounded by this protection of the myelin sheath. Now, in the central nervous system, right, in the CNS, this myelin sheath has a different name and it has different composition as opposed to PNS, right, peripheral nervous system. So they are distinct. In the CNS, these cells, the Schwann cells, are called oligodendrocytes. And in the peripheral nervous system, these Schwann cells are called, these myelin cells are called Schwann cells. So just let me restate it. These myelin sheath in the central nervous system is called oligodendrocytes, and this myelin sheath in peripheral nervous system is called Schwann cells. So why am I making such a big distinction? The reason why is that we have different autoimmune diseases that target different cells depending if they are located in central nervous system versus in the peripheral nervous system. So we're going to learn about multiple sclerosis, which is a demyelinating disease. It predominantly works in the central nervous system. It demyelinates oligodendrocytes. And there's a disease called Guillain-Barré syndrome. It primarily functions in the peripheral nervous system and demyelinates Schwann cells. Schwann cells and oligodendrocytes both insulate axons. They are both myelin sheaths. Their job is to essentially isolate and increase the impulse propagation. So as you see here, this here, they show you here, this axon does not have a myelin sheath. So the way it depolarizes, it goes like from patch to patch. So it becomes very slow. Each patch has to depolarize, repolarize, and only impulse can be propagated patch to patch. So this conduction is much, much slower as opposed to the conduction that's done if you have myelin sheaths and the impulse is propagated with a node of Ranvier. We call this saltatory conduction. So saltatory conduction is much faster, so the response reaches its target site much quicker, right? I'm gonna outline a sensory neuron today, right? Because we did extensive overview of the motor neuron in the muscle chapter, but the sensory neuron, I think it's important to outline so you guys see how that functions. The shape of it slightly differs, right? So think of it like this: the receptor cell can be, for example, it could be like my finger, where someone's applying touch or pressure, right? So if I apply enough stimulus, I'm going to cause my threshold potential to activate, and then the signal can propagate. If the touch is not strong enough, it's sub-threshold. It doesn't reach the threshold. I may not feel that sensation, right? The cell body is in a slightly different location as opposed to motor neuron. It's in the middle here, and then it goes to the target sites. It could go perhaps to central nervous system, and this is your peripheral nervous system. So you feel the touch on the periphery, and then the impulse is propagated through these myelin sheaths, node of Ranvier insulated axons. So the impulse goes faster, but you need enough stimuli to go beyond the threshold potential, right? So this was something I was explaining in the lecture, but I think for the problem sets that I posted, you need to have a good idea of all the stuff. So I'm gonna schematically outline this here, right? So imagine I'm gonna take a sensory neuron we saw, and I'm gonna make a schematic of it. So we're gonna say this is your central nervous system, and here we're gonna say this is your spinal cord. And from here, we're going to take the sensory neuron, right? We see the cell body here. It is just like that we're going to say here it's touch receptor. So this is your mechanoreceptor. This could be your finger, for example, and someone is applying a light touch to your finger. And as the touch increases, we'll see how your signals become stronger, right? So first, very important thing, I said cells are bags of potassium. So we have high content of potassium inside the cell. And we said that the sodium is predominantly found outside the cell. And that was a concept you guys really need to remember, because that is going to be general for most of the cells you're going to learn that have action potential. By the way, can anyone tell me what two cells in the body, what cell types or tissue types have action potential? Action potential means you have depolarization and repolarization.
Cardiac, muscle, and neuron.
And neuro, right? So yeah, so you have, if you want to generalize it, skeletal muscle, cardiac muscle, which is essentially muscle. So muscle and neuronal cells, neurons, right? They have action potential, means they can exchange sodium and potassium ions to take the membrane from resting state, right, to depolarization state. So we know the resting state is negative here, right? And it's roughly about negative 70 millivolts inside. Now, these channels that I drew here, these are not voltage gated channels. These channels that I drew, they're leaky channels. So some potassium can freely equilibrate, it can leak out, right? And this sodium here, this could be your touch receptor channel. This could be your mechanoreceptors. So what that means is that if I apply some pressure to this receptor, some sodium can trickle inside from the outside, right? The sodium can come in. Now, something that's very important to understand, there's different concentration. So here, let's look at extracellular environment. So extracellular environment means the environment outside the cell. And we see the amount of sodium found in extracellular environment is 145 millimolar. Milli is units international, so it's 10 to the negative 3, right? And what's molari? Molari is moles per liter. So this is how the units you see on, let's say, a patient's blood work, right? How do they determine it? So it's millimolar, and moles is molarity is determined by moles over liters. Now, I said extracellular sodium is 145 roughly, right? Millimolar. But if we look what's inside the cell, right? Intracellular. So intracellular is environment inside the cell. It's found in the cell itself. You notice the distinction, right? Only 15 millimolar. So outside was 145, but internally it was 15 millimolar. So naturally sodium wants to come in, right, through its gradient from the outside to the inside, because if you learn about the process of diffusion, right, it's from high concentration to low concentration. So we see that. The second ion that I want to touch upon again here is the potassium ion. And we see inside the cells, the amount, outside the cell, extracellular environment of potassium is 4.5 millimolar. So potassium here outside is 4.5 millimolar. But potassium internally has a different amount. So inside the cell has 120 millimolar, right? So put this here. So this is why I was saying cells are bags of potassium, right? Cells are bags of potassium in the sea of sodium, because your amounts differ. And in the other lecture, I said when you guys get called for your emergency call in the nursing home for abnormal labs, usually what happened, they hemolyzed the cells. They probably shook the tube with blood, and cells broke, and the contents of potassium leaked out. So when they look at the potassium level, it's like 20, it could be 10 or whatever. So obviously at that level, person would not survive. But that's why they call you abnormal labs, because they have to be retaken, right? Because cells hemolyzed internally. Now, besides the concentrations here that we saw outside and inside, there's something else that's crucially important to understand. This is called equilibrium potential. So equilibrium potential being, if I take a voltmeter, right, like a meter that can measure volts, and I look what's inside versus what's outside, right, for these potentials. And what I'm gonna look at is when the channels, right, when those voltage gated channels, right, because here we're looking at the voltage potential. When the voltage gated channels are open, what the cell wants to do. So look at the potassium, right? Potassium here, its equilibrium potential is negative 88 millivolts. So for the potassium, it's negative 88 millivolts. So that means when the voltage gated channels for the potassium open up and the potassium can freely come out from the inside to the outside, it's rushing to reach its equilibrium potential at negative 88 millivolts, right? And when we have sodium channels that open up here that are voltage gated, right? So this is voltage gated, right? Sodium wants to come in from the outside to the inside, and what's the amount for sodium? We see it's plus 61. It's at the top right here, right? So plus 61 millivolts. So as I was drawing the diagram in the muscle chapter, it was way simpler, but here it becomes a little bit more involved. Why? Because now we have to actually see how these channels respond and what that all means, right? So imagine if I drew this graph, right? Let me make these gradations. So at the very bottom, I'm going to draw voltage. So the very bottom is going to be negative 88, and that's the equilibrium potential for potassium. So we said minus 88 for potassium. The next one that I'm going to draw is the resting state. So this is the resting membrane potential, and it's roughly negative 70 millivolts, as I guys told you, right? On the neuron itself, so negative 70 millivolts. The next one I'm going to draw is going to be our threshold potential. So this is our threshold potential. This is when sodium channels will open up, and it's roughly negative 50 millivolts, right? And this is where it's going to occur. And then from here, right, as the cells, let's say, becomes more positive, let's say this is minus 30, this is minus 10, then we have, let's say, 0, this is plus 20, right? It's going to go up until it reaches, we're going to say at this point, this is going to reach plus 61, and that's going to be equilibrium potential for sodium, right? This is what we said, plus 61 for sodium, right? So this is our table. This is what we have. So if I have a stimulus that's coming in, let's say someone's touching my finger and it's not strong enough, right? So from the resting state, I may only go this far. So I have not reached my threshold potential. So do you think my voltage gated channels that are found on this membrane, right? These are the voltage gated sodium channels. Do you think they will open if I have sub-threshold potential?
No.
No, right? They will not be affected. Now, what may happen is you may have a stimulus that comes in. Let me do it like this. You may have a stimulus comes in that right before it dissipates, someone else causes another stimulus. So they're like summative in nature, right? So they're graded, meaning this was low, but then another one comes in, maybe it's a little bit higher, and then another one comes in, and before it terminates, someone touches you again, and it reaches to the threshold, right? So multiple stimuli that are added together can reach threshold, or you may have a strong enough stimulus right away, right? Somebody touches your finger with enough pressure, but with enough force, they could right away cause this action potential to reach threshold potential. And we said negative 70 to negative 50, so we see the membrane is going towards the positive side. So the moment this here occurs, what happens is these voltage gated channels right here, these voltage gated channels, they will open. Before they were in the resting state. The resting state is looking like this, right? You have activation gate that's closed, and you have inactivation gate that's open. But they're in the resting state. But the moment threshold stimulus is applied enough to get to -50, the activation gate opens, and inactivation gate begins to close, but it's still at this point, it's open. And you could see all the sodium is rushing in. What's extremely important to understand is once you reach threshold, it's all on our response. It's not like a few sodium voltage gated channels open, a few don't. All of them open. And there's thousands of sodium voltage gated channels on the cell membrane. So the moment the threshold is reached, we say it's basically the response is all or none. So it's all because your threshold has been achieved. And the moment this occurs, your resting state sodium channels become active state. And the moment active state is achieved, you could see a lot of sodium can come in. Why does the sodium come in? Because first of all, outside the cell, we said there's 145 millimolar. Inside the cell, it's only 15 millimolar. So by the gradient, they want to come in from the high concentration to low concentration. But there's another important factor. What's the other important factor? If we look at the equilibrium potential, right? We said equilibrium potential for sodium is plus 61 millivolts, right? So it wants to reach its equilibrium potential, where I got these numbers, right? Plus 61 is from this, right? That they did experiments, and this is what they derived. So now what you see is the following thing happening. The moment threshold is achieved, sodium will rush in with excessive force, going all the way to plus 61 equilibrium potential, but it does not reach it. It does not reach it. Why? Because we said here the sodium channels were open, right? And the sodium was coming in. But right before it reaches its equilibrium potential, what happens to sodium channels? So what happens is activation gate is still open, but an activation gate shuts. So an activation gate shuts, so sodium no longer can come in, right? We can see that right here, right? So no more sodium can entrain, no more sodium influx can occur. So the membrane does not actually reach fully towards the sodium. It wants to, but it doesn't. And what also happens at this point here, we have voltage-gated potassium channels, right, which are found on the cell membrane, and they become activated. They go from resting state, right? This was resting. They become activated. So activation gate is open, and then potassium can start leaving the cell. Why does the potassium want to leave the cell? Because inside the cell, potassium is 120 millimolar. Outside the cell is only 4.5 millimolar. The other thing is it wants to reach its equilibrium potential, which is now 8 minus 88 millimolar. So there is concentration gradients that facilitate the exchange, and there is also voltage gradient that facilitate that exchange. So now what you see is at this point, right, we stopped here. Sodium channels inactivation gate closed, right? So no more sodium can come in, but potassium is leaving the cell. So if the potassium positive charge is leaving the cell, you could see it's starting to go down, right? It's becoming, right? It's starting to repolarize, right? And it's going down, down, down. Why is it going down? Because it wants to reach its negative 88 equilibrium for the potassium, and it's going to go towards it. But right before reaching its equilibrium potential, same occurrence occur, right? Even though the activation gate opens, the inactivation gate shuts, so that no more potassium can escape, right? And on our cells, on the neurons, there is sodium potassium ATPases. They are found in these pockets all over the cells, and this is sodium potassium ATPase. And what its function is, it's using ATP in order to kick out 3 sodium and bring in 2 potassium. So this mechanism is what will essentially equilibrate the potentials back to the resting state, right? By throwing away back the sodium that was rushing in, and then bringing back the potassium that rushed out to restore the resting state potential. So it will bring this point back to the resting state. At this resting state, sodium channels return back to their normal configuration, right? This is closed, activation gate is closed, and this is the inactivation gate, which is now open. This is the resting state, right? This was the same state that was found here before depolarization was occurring, right? So we saw the sodium channels go through the following sequence. This was resting. The moment we achieved threshold here, it was active, right? Sodium was coming in. At this point, right, it became inactive. Why it became inactive? Because the activation gate was open, but inactivation gate shut. But until this sodium channel returns back to its resting state, no impulse can come in, no impulse can depolarize. That's why we see, right, this state up before you reach up this point, this is called absolute refractory. So all of this is absolute refractory period. Why? Because no stimulus, regardless of the strength of it, can initiate another action potential. But the moment sodium returns to this configuration here, new action potential can start. Again, if you touch my skin here being the mechanoreceptor, right? This what's going to happen. So in this cells, what happens is there's a lot of patches like this that I drew here with voltage gated. They look like this, right? The patches, and they go all the way, right, to the central nervous system. And as you could see, let's say this patch 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? The impulse propagates like this across all the patches for the depolarization signal to reach its destination. Now, what you want to remember, the way this works is the moment this, let's say this cell is depolarizing, this cell is going to repolarize. So this is depolarizing, this is going to start its repolarization. This is depolarizing, it's going to start its repolarization. So you see, right, it's going from patch to patch, causing depolarization, repolarization, and as the signal propagates, right, it changes, it sweeps across the membrane, becoming positive, right, and then it becoming negative again, resting state, and so forth until it reaches its intended site. Now, in the lecture, I asked, right, imagine I take a big hammer, right, and I hit your hand, right? So it's obviously going to be mechanoreceptors. It's obviously they are activated. So my question then becomes, do you suppose, right, your action potential all of a sudden, if I hit your hand with a hammer, looks like this, right? Much bigger, or do you suppose it looks like this? So 1 or 2. So is it becoming more in amplitude or becoming more frequent? Frequency, yeah, very good. So if your frequency increases, and that's what sends more impulses. Your amplitude will not increase. Why? Because we cannot truly exceed those equilibrium potentials that I already drew here, right? You see the chart, right? So the equilibrium potentials will usually maintain the voltage. So you will have more frequency, you will not have more amplitude. The other important thing here I drew an unmyelinated neuron, right? Non-myelinated. So I asked in the other class, but I will ask it in here, right? Let's say I take a cross section, I cut here, and I take a cross section of this neuron, and then I drew 3 neurons. This is neuron number 1, 2, and 3, right? And then this is your nucleus here, cell body. And then we're going to say here we see the following myelin sheath like this. And here we see myelin sheath that forms multi-layers. So we have neuron 1, neuron 2, and neuron 3, right? Out of these 1, 2, and 3, which neuron is myelinated and which is not?
3 is myelinated, 1 is not.
1 is not, right? 3 is myelinated, 1 is not. Any alternate answers? Anyone want to give any alternate answers? Or everybody believes 1 is not myelinated and 3 is?
3 is myelinated and 2 is non-myelinated.
Okay. So Brian is correct, right? So number 3 is myelinated because it's forming multi layers so that there is no leakage of any potassium, sodium, right? Here, I could still have leaky channels that coming in. So all neurons in your body, they all have some form of myelin sheath around them. It may not be fully enveloping the neuron. So meaning there's some opening where exchange can occur. This here is a disease process. This is a demyelination. So this could be Guillain-Barré, this could be multiple sclerosis. So this neuron is diseased. This has a disease process. So most of your neurons in the body have some form of myelin sheath, right? But they may not fully envelop. Those that are fully enveloped, right? We say these have full, surrounding myelin sheath, and this essentially will cause that saltatory conduction, fast conduction, right? Very good. So I will just draw a little schematic to guys show you. So if this was my neuron, right, and it was going to the central nervous system, what these sheaths do, they basically form cells like this surrounding it. So that no sodium and potassium can leak out through here. They can only leak out through these nodes, right, or through these nodes. And this is why the impulse is traveling like this. It's traveling much faster, right? These nodes we call nodes of Renvier, where actual exchange is occurring. So this was the predominant thing that I drew. We talked about the sodium channels, potassium channels. We talk about the myelin sheath, right? One thing that I wanted to show you guys so that you will see. So if this was our central nervous system, brain, and the spinal cord, and here, let's say we had a neuron that's coming in, right? On the peripheral nervous system, right, the PNS, on the PNS here, the neurons look like this. They have the myelin sheath that's surrounding them, which is called Schwann cells. The Schwann cells, they are only surrounding one neuron, so only one neuron. But in your central nervous system, we're going to say your neurons run like this. So this neuron will synapse here, right? So in your central nervous system, right, let's say this is one neuron, this is second neuron, this is third neuron. The way oligodendrocytes work, they will surround multi-neurons, right? And it'll be another extension. So this will be oligodendrocytes. And so the difference is this: even though the function is to sheath, right, provide the sheath, the fat sheath, and isolate the neurons, you could see how the structure and composition slightly differs between peripheral nervous system and the central nervous system, right? This here is being central nervous system here, and this is being the peripheral nervous system. That's why different demyelinating diseases, right, Guillain-Barré and multiple sclerosis, can affect different cells. Guillain-Barré is peripheral, right, peripheral demyelinating syndrome, and multiple sclerosis is CNS, or central nervous system demyelinating disease, right? So this is what you want to be cognizant of, right? Any questions about this mechanism that I drew about the action potentials, the gradient differences?
I have a question about the action potential. So why does the potassium, why do the potassium channels wait for the voltage to go up? Like, is there a specific reason why they don't activate earlier?
Yeah, so they exactly, they have to reach their own threshold. So this was threshold potential for the sodium channels, but threshold potentials for potassium can be way higher, right? So each type of voltage gated channels have their own threshold. For you to activate them, you must reach that threshold. The moment you hit threshold, they all open up. So that's why we have different potential for calcium. We have slightly different potential for potassium, slightly different potential for sodium. But what's critical here is understanding that the two cells that are able to do this is neuronal cells and muscle cells. And predominantly how this is done by sodium and potassium. And this is done by sodium influx, and it's done by potassium efflux. So potassium leaving and sodium entraining. So this is the predominant structure.
Thank you.
Yeah. Something else that's going to be important, right? In the muscle, I said in the setting of hyperkalemia, if you have a lot of potassium, right? Imagine we said normal is 4.5 for potassium, right? Outside the cell. But let's say for the argument's sake, and it becomes, instead of 4.5, it becomes 6 to 7, right? So now you have millimolar. So you have elevated potassium outside the cell. So if you now have more potassium outside the cell, right? Let's say this gradient is no longer 4.5. Do you think potassium wants to come out just as easily as it was 4.5? Or it's a little bit harder because there's higher amount of potassium outside?
Harder.
Yeah, it becomes harder. And what happens is, because it becomes harder, less potassium is leaking. So what happens, it takes its resting state maybe of the cell a little bit higher, maybe it becomes minus 60 millivolts, right? And you would think, oh, it's closer to the threshold, so it will activate faster, but not so. Actually, what potassium hyperkalemia does to these sodium channels, it inactivates them. So instead of being in this resting state where this inactivation gate was open, this high amount of potassium does this, it closes this gate. So this sodium channel becomes inactive, it becomes like this, with activation and inactivation gates closed, so that you cannot trigger the next impulse to propagate. So that's why in hyperkalemia we see heavy muscle weakness. And why do you have heavy muscle weakness is because high amounts of potassium do not let these potassium leaky channels to function well. Less potassium is leaving because there's a high concentration of potassium outside. This renders sodium channels ineffective by closing the inactivation gate here, and the next depolarization cycle cannot occur. So in setting of hyperkalemia, this is dangerous, right? And we'll talk about, once we get to the cardiac chapter, right, some of the potential treatments. So the whole graph that I drew is essentially this graph here. In terms of your starting voltage and your threshold, this will vary from cell to cell. So cardiac myocytes will have their own resting state, slightly different numbers. Muscle cells have their own. Neurons have their own. The actual numbers is not so crucial to understand right now. What's very important to understand is this process of depolarization, repolarization, and this process of absolute refractory period that I talked about, where no stimulus can come in, right? But what happens in the refractory period? In the refractory period, right, the sodium channels has now regained its resting state, right? This was the resting state. And at this portion here, right, we said the sodium channel, even though the activation gate was open, this shut the inactivation. So no sodium was coming in. So what you want to be clear about is that in absolute refractory, no stimulus that you do will activate it. But in relative refractory, right, you can cause depolarization to occur if this sodium channel, voltage gated sodium channel, returns back to its resting configuration. All right. So this was Guillain-Barré. This is demyelinating peripheral nervous system disease. This was multiple sclerosis. It's demyelinating of the oligodendrocytes, the cells here, right? What causes this? B cells, right? Your autoimmune, your immune B cells secrete inappropriate antibodies. They're inappropriately targeting these myelin sheath, specifically oligodendrocytes, to cause them to break apart. Right. So this is what happens in those disease states. The effects, you'll have muscle weakness, ataxias, things of that nature. If it reaches your respiratory system, you may become, right, you may have an inability to breathe because your respiratory muscles are not working. So in the hospitals, the treatments for example, Guillain-Barré, they'll place them on the ventilator so it can breathe for them. They'll give them plasmapheresis to clean out their plasma from bad antibodies, their own bad antibodies that's destroying that myelin sheath, the Schwann cells. And they're going to give them immunoglobulins, the good, healthy antibodies. So hopefully that doesn't occur. And then after a while, right, the body recovers, the myelin sheath gets remade, right? The body remakes the sheath. The person regains control of breathing, the muscles, and they get off the ventilator, weaned off, and get discharged from the hospital.